Muscle tissue is made up of cells that contract and shorten. The muscle tissue allows movement. It also supports the organs. The cells of the muscle are known as myocytes. The meaning of sarco is flesh in Greek. Hence, the cell membrane, cytoplasm, and endoplasmic reticulum are known as sarcolemma, sarcoplasm, and sarcoplasmic reticulum. In this section, we will look at histology of skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscle is in the form of myocyte or muscle fiber. Structurally, skeletal muscles have long cylindrical fibers. They can be as long as 30 centimeters. In reality, it is made up of many myocytes. It is called syncytium of myocytes. For the same reason, it is multinucleated. Nuclei are flat and they are arranged peripherally within the muscle fiber. They are pushed peripherally because number of myofibrils are occupying the core of the muscle fiber. This is part of the skeletal muscle fiber showing eccentrically placed nucleus. Inside the muscle fiber, we see many myofibrils. Every myofibril shows dark and light bands. All the dark and light bands in adjacent myofibrils are perfectly aligned. This gives banding pattern on the muscle fiber also. These are called striations. We will see the banding patterns in detail. All the dark bands are formed by protein myosin. All the light bands are formed by the protein actin. In the center of the light band, we see Z line. We shall see the details. Dark and light bands are shown with color differences. These are dark bands, dark brown colored, and in between, light bands. Within the dark band, we see myosin filaments. And within the light band, we see actin filaments. Myosin are thick filaments, actin are thin filaments. Dark bands are anisotropic bands. Light bands are isotopic bands. That's why we call them A band and I band. And in the center of the I band, we find Z line. Z line is the one which holds actin filaments on either side of it. And in the center of the dark band, we find M line. M line is the one which holds myosin filaments on either side of it. The part of the myofibril between two Z lines is called sarcomere. Sarcomere is structural and functional unit of muscle fiber or myofibril. In the area of light band, if you take cross section, we will see only actin filaments. But in the area of dark band, the peripheral part of the dark band shows overlapping of myosin and actin both. But towards the center, if you have the section, so that shows only myosin filaments. So this overlap makes dark band much darker at the periphery, and in the center where there is no overlap of actin filaments, this is called H band. During contraction, Z lines come closer. So these two Z lines have come closer and actin filaments are sliding inside the myosin filaments, and the H zone will decrease in its size. These are the adjacent myofibrils showing dark and light bands. All of these are arranged within the muscle fiber. This is the sarcolemma and these are the mitochondria. This is the transverse section. Interestingly, we see some tubular structures inside the muscle fiber. These are triads, 3 tubes are arranged at the junction of dark and light band. We will see what they are. So we can see these. The one central dark blue colored tube is having communication to the surface of the cell, that is sarcolemma. The cell membrane invaginates inside in the form of T tubule, which is going around every myofibril. Just by the side of it, we find terminal cisternae of endoplasmic reticulum. Why this is necessary? This type of arrangement allows the contraction of all the myofibrils within the muscle fiber whenever the muscle fiber is depolarized. Calcium ions are necessary for contraction couple to happen at actin and myosin filaments. Calcium ions are richly concentrated in sarcoplasmic reticulum. The depolarization wave from the sarcolemma quickly disseminates inside through the T tubules around every myofibril, and that's how calcium ions are released and contraction happens in all the myofibrils. We see triad of 3 tubes at the junction of dark and light band in skeletal muscle, but in cardiac muscle we see dyad of only 2 tubes at the level of Z lines. These are cardiac muscle fibers which are short and branching. Here it is branching. These are the cell junctions. Due to infolding of the cell membrane, cell junctions appear as intercalated discs. Here it has tight junctions and gap junctions. Gap junctions make whole cardiac muscle a physiological syncytium. Because of less myofibrils, nucleus is occupied centrally and striations are weakly seen. Smooth muscle cell is spindle shaped. After contraction, it is globular. Thick and thin myosin and actin filaments crisscross the whole cytoplasm. They are attached to the cell membrane at dense bodies. Adjacent cells also show gap junctions, which spread the excitation throughout the visceral muscles. Let us look at connective tissue component of skeletal muscle. Having myofibrils inside and peripherally pushed flat nuclei, every muscle fiber outside to its sarcolemma is covered by delicate connective tissue, that is endomysium. Many such muscle fibers together constitute muscle fascicle. On the outer limit of the fascicle, it is perimysium. Many such fascicles together make up whole muscle belly. Whole muscle belly is covered by epimysium. What is the function of these connective tissue layers? Epimysium is pierced by neurovascular structures which supply the muscle. Perimysium and endomysium allow these branches of neurovascular structures to pass and reach the muscle fibers. These layers also help in transmitting the force of contraction from endo to perimysium, peri to epimysium, finally to the tendon and to the bone for the movement. Histology of skeletal muscle show long, unbranched cylindrical fibers. Nuclei are multiple. They are placed peripherally in the muscle fiber and flat. There are evident striations which are seen because of actin and myosin filaments inside the muscle fiber which are arranged within myofibrils. This is transverse section of skeletal muscle which are arranged in different fascicle. So this is the boundary for every fascicle, and fascicle is covered by perimysium, and every muscle fiber is covered by endomysium, and whole of the muscle belly will be covered by epimysium. In the transverse section, we see nuclei which are peripherally located within the muscle fiber. This is histological picture of longitudinally cut cardiac muscle fibers. Here, fibers are cylindrical, which are short cylindrical, and they show branching. Nuclei are single in every myocyte, and they are central and oval in shape. And in between the myocytes, the cell junctions are shown with intercalated discs. These are intercalated discs. Striations are seen here also, but they are not very evident as in case of skeletal muscle. This is microscopic picture of smooth muscles. This is longitudinal section. These are transverse sections. You can see muscle cells in the shape of spindle. Nucleus is single for cell, and they are centrally located oval. These are the transverse sections. You can see muscle fibers are arranged in different fascicles. Striations are not seen here. That's the reason they are called smooth muscles.